Hi guys, welcome back to my channel. I'm Ananya Rana and today we are going to have our project submission. So guys, let's get ready for it. Our project is being made right now and we have kept our project in gym room. Here is it guys. See, in the working conditions, we have finally started about it. Here's the thermocol. Here is all the products that we are going to need later. And our topic is electron transport chain. So guys, it's going to be made today and then projected tomorrow in the lobby. So this is the discussion of today's video. Hit like, bye bye. Hi guys, welcome back to my vlog. Today we are going to have our bio display. Like last time I recorded my physics subject exhibition. This time I'm going to bio exhibition with in my team this time. So we take the chart paper.
Oh my god! Wow!
Yeah, you have to go back. Okay. She's going to get the chart paper right now. I'll get to the class because we are getting late. So yeah, we were given one responsibility, that was to get the chart paper ready, and we left it in the room. Anyways, we are not late for the exhibition, so it doesn't even matter though. We are going to the class. Let's go. See, they are getting their projects ready. I thought I am the only one who's going to be late. This is Sarah and this is Vasudha. What is your project this time? DNA replication. Our topic is ATP synthesis. Yeah. Okay guys, we are getting our attendance done. I am done with my attendance. And this is Shazia. What is your project today?
Electron transport chain.
Yes, we have to leave for the bioengineering block later for the project exhibition. It's biochem. It's biochem right now, and we have cell bio later. She's going to leave us soon. Can you hear her? Just make this reader. See, this is Adi, this is Adi, and that is Charu. She's the captain. He's playing with the gun, I don't know why. We are working on the project. Yeah, they are getting their project really ready right now. See, stealing the spotlight, that is very nice. We can just leave. It's not even been half an hour since we were in the class and we can just move. We are going to the hostel and then we'll get the project and then we'll go to present. Guys, in my block. Yeah, these are my teammates. This is Aditi and this is Tanisha. You have seen her before in my vlogs. Not coming. And she is the left lady today.
No ma.
No, okay. Ma, MS press MS ma.
No ma, once only 11th floor.
Said that there's no point going too early, so we'll stay in our hostel.
Huh?
Me, yeah, we'll stay in the veranda.
We have our model ready. Yeah, within one day product. So we are so efficient.
Putting a shawl over this thing. Like, grand reveal. Guys,
Oh my
grand reveal.
god.
अरे ये वाला मेरे को ब्लर कर दो.
इतना नहीं है मुझ में टेक्निकल स्किल्स. our project. Guys, you know what? Our project is very mindfully made, very thoughtful. I'll show you how. So here is our project. This is electron transport chain. This is complex 1, 2, 3, 4, and this is 5. Zoomed in 5. So I know what is the working of complex 5. Charu, please do the favor of explaining these parts.
This happens in inner mitochondrial membrane. This entire process happens in inner mitochondrial membrane. We have complex 1, complex 2, complex 3, complex 4 and complex 5. Our main focus is on complex 5. These are like secondary to explain the prerequisites. So complex 1 says we have NADH2 comes from the Krebs cycle, and we have FADH2 which is the electron carrier. NADH2 comes, it gives 2 electrons to FMN. FMN gets reduced into FMNH2 and provides electrons to FES. FES transfers electrons to UQ, as you can see. Now this UQ gets reduced to UQH2. Let's talk about this later. Ab chalte complex 2. So complex 2 basically provides what? FADH2 provides electrons to complex 2. This FADH2 gets reduced into FAD. And this gets oxidized into FAD, and this FAD gives its electrons to FES. This FES provides its electron to UQ. So basically both complex 1 and complex 2 provides their electrons to UQ. Here, एक और बात which we should know that this simultaneously as it's providing its electrons to FES, the protons are also being pumped consequently, and then what happens? Complex 2 does not pump any proton. So both of them provide their electrons to UQ. The UQ gets oxidized into UQH2, reduced into UQH2, and then UQH2 comes to complex 3, gives the electrons to FES, FES gives the electrons to cytochrome C. Now this cytochrome C travels and goes to complex 4. So this cytochrome C travels over here and gives its electrons to copper, copper gives it to cytochrome A, then cytochrome A gives it to copper again, and here is the main thing which happens. Oxygen is waiting over here. Oxygen binds with the protons, and as oxygen binds with the protons, it produces water. We have oxygen which is the final electron acceptor. So here water is produced, and then and consequently protons are also being pumped. So we have covered F1, F0, which Ananya will tell.
So here, this is F0 and this is F1. F1 has 3 subunits, alpha, beta and gamma. 3 pairs of these subunits. We have 3 subunits in the F0 also, alpha, A, B and C. उसके बाद यहाँ पे हमारा प्रोटॉन ग्रेडियंट है, जिसकी वजह से जो प्रोटॉन होता है वो F0 में आता है. F0 के अंदर हमारा जो C subunit होता है, which it has a residue which binds the proton, which binds the proton in this way. And we have A, which allows the exit and entry of the proton. We have B, which restricts the rotation of F0 but allows the rotation of C ring. Now what happens is the proton from this goes down. यहाँ से निकल जाता है प्रोटॉन, because of which the C ring is rotating. Once the C ring is rotating, the gamma here XL also rotates, लेकिन धीरे धीरे रोटेट होता है. इसका जैसे 3 रोटेशन होगा, एवरी 3 रोटेशन. नहीं, ये नहीं बोलना है कि गामा का जब 3rd रोटेशन होता है इन द सब यूनिट्स,
When there is a 3rd rotation of gamma in the subunits,
ADP is converted into ATP.
okay.
Hi guys. Say hi. Do you care to explain the model, Hazel?
Hold on, please. One second. I will surely explain this, but just give me 1 minute.
Yeah. Please care to explain the model?
She is the one who is going to explain us.
Oh my god. Hi, Ragalda. Left. Oh wow.
Oh no.
Oh, they're sitting there.
What was your explanation?
This is ATP synthesis and electron transport chain. So done? Yeah, we're done.
You look pretty good. I have the lipstick on. Just because of the video.
It doesn't matter. How do what we have this bioengineering. Otherwise, we have open
this
sky.
plays the entire
College, yeah. Otherwise, it feels like a
school. Typical school. 8 to 5 classes.
8 to 5 classes, and then we are not even allowed to have lunch. You can't have this, you can't have that. Can you explain your model today? Explain karo.
Explain. This is DNA replication. So this is DNA replicating structure. This is helicase. So basically it is a working model.
Oh my god!
So initially it was here and these strands are closed.
Wow.
Whereas because of the helicase, the strands basically start
Cutting.
splitting.
Splitting.
Slicing, you can say.
Yeah.
It comes here. And we show how the moving of the sequence.
Sorry, what?
We showed the moving of the sequence. This is how the sequence starts. Our sequence is stuck, guys. Malfunctioning, but okay, it's fine.
It doesn't happen in the DNA though, guys. DNA is very smart.
DNA is nice. Your
DNA is nice. You don't have an error in that.
No error. This model DNA is very obedient. No glitches. Work properly.
No errors. No errors. I'm so scared now because I think I am getting too much into others' project that I will forget my own project. Again, see? The project. Your project is
Mitochondria, structure of mitochondria.
mitochondria. Mitochondria is the power of the house.
Most famous topic of
Bio.
mitochondria is the power of the cell.
That's all.
On the lights once.
Guys, this is also a working model. See? Oh my god. Efforts. After learning mitochondria, you can come there and learn the electron transport chain.
यहाँ से वो डिटेल में बताएगा
हाँ.
हम लोग को माइटोकॉन्ड्रिया ठीक है तो इसी को डिटेल में एक्सप्लेन करेंगे इलेक्ट्रॉन ट्रांसपोर्ट चेन क्या है
Yeah. That's way.
Pathogen enters, or like if pathogen is found in a cell, phagocyte reacts to it by it recognizes the pathogen by opsinin. So and it makes pseudopodia-like structure to engulf this pathogen. And when engulfed, membrane-bound vesicles and bind to the pathogen, making a membrane-like structure. It's called phagogen.
Oh.
And this phagogen.
I mean, he's asking the questions. I'm so scared of. Yeah, the professors are good, so they will give us. They would, right? They would, right? That's what we're hoping. We will get full marks, right, Charu? Yeah. Yeah. Okay, they're done with their project. So let me ask them. You want to see my CR? We have to both the CRs in the project. Wait. He's the first CR. And not the first CR, he's the CR. Olivia, see the CR. CR, that's the introduction of yours. Are you done with the project? Yep. How was it?
Nice. But asking a question which I didn't use. I think I knew, but it was like I know that she but I don't know how to explain it. The usuals.
No, see, usually asking tough questions, so just pointing out and answering.
Easy questions. Bro, you're done. What was he asking?
We have two topics. Having two topics.
Easy question. He'll just be like, Ananya, explain this after seeing the model. Like, he'll not even think that that's not a topic, that's not inside. What is your topic? I was neurons and he asked about like brain cells. He asked about brain cells. Why? What is your topic? He told me the answer. I don't know neurons. I think even he's not giving marks. But if he studied whatever, he would not have answered that he's not giving marks. So he's not asking anything related to the project. He asked asking, but on the why it explained to explain.
Why it first? We were looking so good.
Exactly, I just felt it when you entered. And then your dark lipstick was looking much better than it was.
Thank you so much.
Joy of
Hi. Ananya.
that's exactly my reaction. That was my reaction.
See, what I'm more scared is that I just don't know the electron transport. I just know how the synthesis work. I know the whole thing, everything in that. But I don't remember the process of electron transport. Sir.
And we can ask you that.
Guys, the question era was not the best part to be recorded because, well, we were all scared about it. We had the worst question asked, but we managed to just pass it anyhow. How was your experience after answering the...
It was fine. My question was comparatively easier, I think.
Yeah, I had this, why is your chart not having this thing? And I was like, okay, yeah, sir, that was the thing. Okay, wait, I'll show you something. Guys, Ashima is also having her exhibition right now. I wish I can just take a photo of her so it's possible. Wait, I'll just try to get a glimpse of her in order to say like, yeah, she did something, so yeah. Let's go to Ashima before anyone else goes. प्लीज से मत करा दे हमारा स्टार्ट हो गया था ओके बाय भाई मैं निकल रहा हूँ guys I think her teacher is a bit of a strict teacher so I cannot go and talk much to her anyways I think I left my classmates I mean teammates yeah they are not where did they go? You know what? I was so scared. He just said, Ananya. Yeah, right? I was doing ChatGPT at that moment. What is the thing? What do you understand? He was like, just speak. What do you understand? What? He asked you what was your part in this project? I was like, वो गए क्या छोड़ के सारे बेस्ट फ्रेंड है बेस्ट फ्रेंड वाओ क्या आधे से ज़्यादा पूरा दिन ऑलमोस्ट पूरा दिन गए सब तो पता क्या
24 घंटे
बस सोने के टाइम पे हम लोग अपने अपने रूम में होते हैं मतलब सोने के भी 4 ही तो घंटे होते हैं
4 ही घंटे होते हैं उसमें भी कई बार हम नींद में एक दूसरे के रूम में जा रहे हैं
हाँ कि भाई उठ जा ये कर लो एक मिनट तो मेरे क्लास के लोग मुझे छोड़ के चले गए ओह दे आर लर्निंग वेव बोर्ड यू नो व्हाट माय कजिन used to have a wave board and she used to learn and my sister also knows how to do the wave board thing I don't actually you know how to do wave boarding Shazam